हेलो गाइस, आई एम शुभम फ्रॉम शुभ ट्यूटोरियल्स। हमने ट्रांसलेशन के पीवाईक्यूज देखे इन द पार्ट 1। वी विल कंटिन्यू विद द क्वेश्चंस इन दिस वीडियो। ओके, so before we start, subscribe to our channel and press the bell icon to get notified whenever the video is out. This is absolutely free from your side, but it will help us grow. First question: during each cycle of chain elongation in translation, how many conformational changes does the ribosome undergo that are coupled to GTP hydrolysis? Okay, so translation पूरा हमने discuss किया है in the molecular biology, right? Very important topic it is. So this is about the elongation process, right? Elongation process. So basically elongation process में first जो amino acid tRNA है A site पर आता है, you know that, right? तो ये हैवी मॉलिक्यूल है, एज आई सेड टू रिमेंबर, हैवी मॉलिक्यूल है तो इसको एनर्जी चाहिए ए साइड पर लाने को, तो दिस आल्सो रिक्वायर्स जीटीपी, राइट। एंड जो ट्रांसलोकेशन का प्रोसेस है विच इज गेटिंग अहेड ऑफ द कोडॉन, राइट। सपोज दिस इज अ कोडॉन 1 एंड दिस इज अ कोडॉन 2, राइट। सो यहाँ से यहाँ शिफ्ट होना है, okay, translocation that is said. So that process also requires GTP. So basically two molecules of GTP are used, okay, so that is coupled with GTP hydrolysis. So answer is two molecules, right? Conformational changes यहाँ पे पूछे हैं. Which one of the following RNAs possesses the peptidal transferase activity? Okay, peptidal transferase means what? Peptide bond formation, right? जो पेप्टाइड को ट्रांसफर करते हैं, तो that is present in 23S rRNA in the prokaryotes, right? And 28S rRNA in the eukaryotes. Okay, this is also very important and repeated question. A protein has 4 equally spaced trypsin sensitive sites. Okay, trypsin sensitive sites है equally spaced, which results in peptide fragments A1, A2, A3, A4 and A5, right? Means what? Suppose I draw a protein here में 4 trypsin sites है, right? So this is the first site, second site, third site and fourth site. Okay, this gives rise to 5 peptide fragments A1, A2, A3, A4 and A5. Okay, so not there in this order, right? ऐसे ऑर्डर में नहीं है वो प्रोटीन्स आफ्टर डाइजेशन विथ trypsin. Peptides A2 and A5 represent N-terminal and C-terminal fragments. ये दिया है क्वेश्चन में, so I will erase this. As we know, ये N-terminal होता है and ये C-terminal, right? So ये A2 है, okay, A2 है जो N-terminal है and A5 is C-terminal. तो बीच के जो यूनिट्स है हमें पता नहीं है, राइट? इम्पोर्टेंट, इसी से क्वेश्चन सॉल्व हो जाएगा. आई विल शो यू हाउ respectively. Now you are asked to synthesize this protein. Okay, synthesize करना है प्रोटीन का at time t is equal to 0, मतलब स्टार्टिंग में you added all the 20 amino acids, right? 20 amino acids हमने ऐड किए, labeled with 14C, कार्बन को लेबल किया है and initiated the synthesis. Okay, at time 0 this is happening. Then at time t is equal to 4, full length protein is synthesized. ओके, फुल लेंथ प्रोटीन सिंथेसाइज हुई. इफ यू स्टॉप द सिंथेसिस ऑफ द प्रोटीन इन टाइम इज इक्वल टू 1. सी वेरी इंपॉर्टेंट. अभी टाइम 1 पर प्रोटीन सिंथेसिस को स्टॉप किया है, टर्मिनेट किया है, एंड डाइजेस्ट द प्रोटीन विथ ट्रिप्सिन. ओके, डाइजेशन किया ट्रिप्सिन के साथ. विच पेप्टाइड विल हैव द मैक्सिमम लेबल देन द अदर्स. ओके, मतलब ये जो पेप्टाइड्स है अभी A1, A2, A3, A4, A5 टाइम इज इक्वल टू 1 पर जब टर्मिनेट किया है प्रोटीन सिंथेसिस को एंड डाइजेस्ट किया है ट्रिप्सिन के साथ, तब मैक्सिमम लेबलिंग विच पेप्टाइड फ्रेगमेंट विल बी हैविंग. कौन से पेप्टाइड फ्रेगमेंट में लेबलिंग हमें ज्यादा मिलेगी. ओके, वेरी बेसिक. जो प्रोटीन सिंथेसिस की प्रोसेस है, इट स्टार्ट फ्रॉम एन टर्मिनस टू सी टर्मिनस. तो एन टर्मिनस पर स्टार्टिंग में एट t इज इक्वल टू 1 पर हमने प्रोसेस को स्टॉप किया है. तो एन टर्मिनस साइड पर ही हमें लेबलिंग मिलेगी. तो व्हाट इज एन टर्मिनस? A2 as given in the question, right? So इसका ऑप्शन होगा ऑप्शन 4, A2. Got it? This is how you can solve the question. During translation in prokaryotes, when ribosome reach the termination codon, okay, ribosome termination codon को reach हुआ है. The termination codon is recognized by the class 1 release factors, RF1 और RF2. Okay, class 1 release factor है, leading to the release of the polypeptide. Now, a second class 2 release factor, RF3, facilitate the termination process. Which of the following statements regarding the mechanism of action of release factor is incorrect? Okay, incorrect हमें पूछा है. So we know class 1 में 2 release factors है, RF1, RF2, and in class 2 release factors में RF3 release factor होता है. Okay, and in eukaryotic case तो ERF1 ही होता है, RF2 is absent, right? So this handles both. Okay, now we will see what are the statements. Class 1 release factors decode the stop codon, right? Stop codon को decode करते हैं, recognize करते हैं basically, while RF3 is a GTPase that stimulates recycling of the class 1 release factors. Yes, जो class 1 release factors है पॉलीपेप्टाइड को रिलीज करते हैं, राइट? एंड जो क्लास 2 वाला रिलीज फैक्टर है वो क्लास 1 को रिलीज करते हैं, बेसिकली टू रिसाइकल देम, राइट? सो दिस इज द करेक्ट. ओके, हमें इनकरेक्ट देखना है, राइट? इनकरेक्ट देखना है. प्री RF3 हैज हायर अफिनिटी फॉर GTP देन GDP. प्री RF3 has higher affinity for GTP. No, this is wrong, wrong statement. ये ही wrong statement. Higher affinity for GDP है because this is bound to RF3, GTP से ये bound है. Okay, when this functions तो उसका GTP hydrolysis होगा, right, to GDP. Okay, so it does not have the higher affinity for GTP, right? So this is incorrect. Next statement, RF1 and RF2 share a conserved segment of GGQ sequence, which is essential for the polypeptide release. Yes, GGQ sequence होता है, that is conserved statement, so this is also correct statement. Next one, RF1 and RF2 individually possess another stretch of tripeptide sequence, okay, which are involved in the recognition of the termination codon. This is also correct. Okay, मतलब RF1 and RF2 को उनके पास और एक specific tripeptide sequence है. That sequence I will tell you now because that I have not included in the original video, right? So RF1 and RF2 दोनों भी recognize करते हैं UAA को. Okay, this is stop codon. Then RF1 recognizes UAB and RF2 recognizes UBA. Okay, specific है. So now RF1 के पास जो tripeptide sequence है specific, that is PAT. Okay, PAT. Proline, alanine, threonine. Right? You have to remember this. PAT sequence है. And जो RF2 के पास है, that is SPF. Serine, proline and phenylalanine. Right? Serine, proline and phenylalanine. Next question. In prokaryotes, the initiator tRNA is first charged with methionine. Okay, we know this. Followed by addition of formal group to the methionine by the enzyme methionine tRNA transformylase. Given below are several statements in this context. All prokaryotic proteins have formal methionine at their amino terminal end. In this question, I will tell you all and none phenomenon science में नहीं होता mostly, right? So all prokaryotic proteins मतलब सब प्रोटीन्स में फॉर्मेटेड methionine amino terminus पर होगा ही, not a compulsion, right? So this will be an incorrect statement. Okay. Deformylase removes the formal group from the amino terminal methionine. Yes, correct है. Right. See, हमने देखा है जो इनिशिएटर tRNA है फॉर्मेटेड methionine होता है, but there is an enzyme known as deformylase जो फॉर्मल ग्रुप को रिमूव कर सकता है. Okay, so this is a correct statement again. Next, all prokaryotic protein have methionine at the amino terminal end. Not a compulsion again. मतलब methionine ही चाहिए एंड टर्मिनस पर, ऐसा नहीं होता, राइट? सो ये भी ऑप्शन नहीं होगा. तो हमें बी चाहिए, बाकी सब एलिमिनेट. ए ओनली कट. बी एंड सी नो, बी एंड सी वी डोंट वांट. लेट्स सी अदर ऑप्शन. Aminopeptidase often remove the amino terminal methionine. Let's check other option also. Aminopeptidase remove amino terminal formal methionine. No, formal methionine को नहीं, methionine को निकाल सकता है. So yes, ये option होगा. Okay, because formal group is removed by enzyme deformylase. Right? So we want option B and D. Option number 4. Got it? Some cells possess peptides which contain D form of amino acid. How do they arise? These peptides are produced by ribosomes by incorporating D amino acids at specific position. No. Okay, I will tell you this directly. Basically, जो L amino acids है ना, वो incorporate होते हैं, right, in the peptides and not the D amino acid. What is the reason behind this? बेसिकली जो एंजाइम है ना, अमाइनो असाइल टीआरएनए सिंथेटिस, अमाइनो असाइल टीआरएनए synthesis. This can recognize only D amino acid. Sorry, only L amino acid. Okay. So how then D is incorporated? Let's see other statement. Ribosome make peptides with L amino acids only. However, some of the amino acids in the peptides are replaced by D amino acids by a pathway that involves excision of L amino acid. No, this is also कुछ भी दिया है. The peptides with the D amino acids are produced in a ribosome independent manner. Yes, this is correct. मतलब ये जो D amino acids का जो incorporation है, that is by ribosome independent manner. Okay. Next statement, peptide with D amino acid exist only in archaea where they are made by the presence of racemase. No, this is also incorrect. Okay. This is how you can solve the questions by using the options given and the concepts you have seen. Right. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel and stay tuned for the upcoming videos. Thank you.